Right now, I am happy to introduce, and I've lost his bio, my friend David Kong, who is a synthetic biologist, a community organizer, musician, and photographer based in Lexington, Massachusetts. He's the director of the MIT Media Lab's new, I don't think it's new really anymore, but the Community Biotechnology Initiative. And their mission is empowering communities through biotechnology. And he's done a ton of things. He's had graduate studies at MIT, he got a master's degree, he got a Ph.D, he's done all this really amazing lab on a chip work. He's been recognized for a lot of things that he's done. But some of the fun things that he's done is also, he's really involved in community organizing and is also a founder of an art, technology, and community space in Cambridge, Massachusetts called EMW. And that organization's mission is to empower communities through the transformative power of artistic expression. And he actually, his work is really rooted in social justice. So you've kind of had a thread today of social justice, and David is kind of using his background in biology and art to really also work in that area. And so the community programs of EMW explore expressive forms ranging from poetry to electronic music to beatboxing to biohacking and more. And David is a very accomplished beatboxer. I don't know if that's part of his presentation. He's also a vocalist, a rapper, a DJ. He said if I told him to bust a move, he would, but I might not, I don't know if those pants can handle it, so we might not do that right now. But he's just really talented in a lot of regards. He showed us at dinner last night how great a photographer on an iPhone he is. Like, he just is amazing. So I will stop talking so that you can hear David's wonderful presentation, but let's welcome him to NOMCON, David Kong.
Thank you. I think I'm gonna, can you guys hear me? Can you guys hear me okay? Can everybody hear me all right? Yes? Yes? Okay, awesome. Before I start, I just wanted to say, can we all just thank Dorothy? Just big, giant round of applause and thank Dorothy. And also, just all of the organizers of this event. Seriously, I think part of what I think is so cool about this event, you are all here, but you all have your own spaces in your own communities, and you know how hard it is to organize. And so, I know for myself, as doing a lot of different types of events and organizing things, you always have to prepare for that crazy thing that's going to happen. There's always something. And I feel like, Dorothy and what you and the organizers have been dealing with, just things that have just occurred throughout this event, and it's just been so awesome to watch you just handle it and just make it work and just deal with it and just do such a great job. So Dorothy, thank you again. You're so awesome. And all the organizers, again, you guys are so amazing. So thank you all so much, really. And yeah, for me, again, this is my first NOMCON. It's a real honor to be here. As Dorothy mentioned, I wear a few different hats. I do, kind of technical work in synthetic biology. So I'm actually curious, how many of you consider yourselves a biomaker? Okay, a few of you. How many of you have actually been in a wet lab before and held a pipetter before? Okay, more of you. That's great. How many of you don't really know what's going on with biology, synthetic biology, the life sciences, but are really curious to learn more? A lot of you. Okay, cool, good. All right, so I prepared some background stuff too to hopefully kind of get everybody on the same page. So my background, as Dorothy mentioned, I'm a technical synthetic biology guy. So I'll explain a little bit more about what synthetic biology is. But my life, I kind of was doing, this work in synthetic biology when I was in graduate school, and at the same time, I was a community organizer and a social justice activist in Boston. And I was doing these two things just kind of in parallel, not sure how those two things converged. And then eventually, and I'll share this a little bit more in the talk, those things actually converged into kind of what I'm doing now today at the Media Lab, but also in a community center that I run in Cambridge that Dorothy mentioned. And I'm also a musician, and I do a lot of stuff in the arts. And again, I feel like making is about not just electronic, mechanical things. It's also about creative things, musical things, poetry, etc. I think there's a whole spectrum of making that I think all of you engage with. So it's a real honor to be here to speak with you all and share a little bit about the work that we're doing. And also this larger movement around kind of grassroots biotechnology and the life sciences, which I'll share and talk about today. So, okay, so let me see, make sure this slide works. Okay, yeah, so this is me again. This is the Media Lab. This is EMW, which is the space that I work in, and I'm on all the social media things, so you can stay in touch there too if you want later. And so basically, Currently, I direct what's called an initiative at the Media Lab. So I don't know if any of you have been to the Media Lab before, but it's a really, really wonderful place. There's around 35 or so different research groups that are at the lab. I think many of you are familiar with Neil Gershenfeld, who directs the Center for Bits and Atoms, which is related to the Media Lab. And so the group that I direct is something called the Community Biotechnology Initiative. And so we do a couple of things. About a third of what we do is actually community organizing work. And then, which I'll talk quite a bit about. And then another third is social science. So we actually study the ways that communities and global groups can actually innovate and do cool things together. So like crowdsourcing, community-driven innovation, stuff like that. I work very closely with what's called the Center for Collective Intelligence at MIT. And then the third thing that we do is actually tool development. So, a lot of the conversations today and over the past couple of days at NOMCON are about accessibility, open source. And so a lot of our group focuses on developing these low cost and hopefully very accessible and open source tools for biotechnology. And so a big part about all of this, though, is really trying to turbocharge this global grassroots movement, which is really kind of exciting. And it's a real honor for me to play a role in this larger movement. And so, just first a few pictures of just some of the wonderful students that I get to work with at the Media Lab. So these are some undergrads and grad students and so on that I get the fortune of working with. And so this is kind of my Media Lab life. And then I mentioned I also run, and I'm the founder and now current board president of an art, technology, and community center called EMW. I'll explain the history of that a little bit more, but we explore all kinds of artistic forms at EMW, including community bio or DIY bio in our space there. And again, a lot of really wonderful folks that I get to work with as part of that space. So those are kind of the two hats I wear. And I want to start by just with a little bit of terminology, because again, I think a lot of you that may be new to the life sciences there are a lot of terms that people use. And frankly, a lot of the community is still figuring out, I think, what a lot of these terms mean. So broadly, right, you can think about biology. There's a scientific aspect of biology, which some might refer to as the life sciences, which includes things like studying the biology of molecules, like DNA, RNA, proteins. That's molecular biology. There's microbiology, which is studying things like bacteria and microorganisms. There's cell biology, which is looking at things like mammalian cells and so on. So that's kind of on the science side. And then there's a whole engineering side, which is much more about making, right, which I think is really what a lot of what this community really cares about. And so there are a lot of terms like biotechnology, genetic engineering, and then something called synthetic biology, which is really about how we make with biology, okay? And I drew a little line here because most of what's happened, almost all of what's happened with the life sciences and bioengineering is actually in the context of either academia or corporations, and sometimes government, right? So this is kind of real traditional institutions. And what's happened over the past 10 plus years has kind of been this other thing that's more connected, I think, to the broader maker movement, which is sometimes referred to as do-it-yourself biology, biohacking, biomaking, citizen science, right? These are all terms that may be familiar to you. A term that we've been kind of playing around with is something called community bio, right? And community bio, I think, is sort of an evolution of a lot of these existing forms of making, but really, in my view, really more connected to a global effort. So not just any individual labs, but actually networks of labs. And so that, I think, hits with the theme of this event, which is really about the network, right? So that, to me, is, I think, one of the really exciting emergent parts of what's happening with this global movement is the fact that it's not about any individual labs anymore. It's about a global movement and how we all work together. So those are some terms. And again, some of you may have seen this slide. So this is from Neil Gershenfeld, my colleague at MIT, where he kind of compares the history of information technologies going from mainframe computers to personal computers to digital fabrication, where we've got kind of large-scale numerical control machines to 3D printers and desktop machines, and then maybe someday the Star Trek replicator. And so one of the questions is, where does biology kind of fit into all of this, right? And so, Nicholas Negroponte, who's the founder of the Media Lab, recently over the past few years, he's been saying, biotech is the new digital, right? And part of, Nicholas is kind of famous for making a lot of predictions about the impact of information technologies and digital technologies. And part of what he's saying is that, biotech is going to be a giant, giant deal, right? It's already increasingly a major part of our economy. There's a larger kind of bioeconomy. And so, but as biotechnology kind of enters our everyday lives, we're going to see it, even more and more profound, the impacts. And so what are we talking about, right? One of the core foundational technologies of what's called synthetic biology, which is, I'll explain a little bit more about in a moment, is something called DNA synthesis. How many of you have heard of DNA synthesis before? Okay, most of you. So, but it's pretty profound, right? So we're literally talking about a machine, a DNA synthesizer, that can take bottles of the DNA bases A, G, C, and T, and basically print arbitrary pieces of DNA, okay? So DNA is the code, quote unquote, upon which, the code upon which all living systems in the entire planet are based off of. And we now have a machine that can kind of print arbitrary molecules of DNA, which means at some level we're trying to learn how to reprogram and engineer the living world. And so, this is just a little kind of figure showing DNA across scales from kind of individual genes to collections of genes, which are sometimes referred to as genetic circuits, which I'll talk about in a second, all the way up to kind of the smallest genomes for self-replicating organisms. So anybody have any idea what the smallest genome for a self-replicating organism is? Anybody have any ideas?
Yeah, I'm a ringer in the audience. How narrowly do you define self-replicating?
Well, how are you defining it? Throw it back to you. What are you saying? Huh?
Pick a virus, 5x174, 55.
Okay, that's a good one. So that's an interesting question of virus. If you want to go with viral genome, I was going more for microorganisms. So the smallest known microorganism that I'm aware of, at least, is Mycoplasma genitalium, which is about 580,000 base pairs, and that's actually been synthesized before. So there's a guy, an institute, the J. Craig Venter Institute. They've actually synthesized like a 6 megabase genome, totally synthetic, okay? So the point overall is that we have developed this increasingly, kind of amazing ability to synthesize these molecules. And then I think all of you have probably heard about CRISPR, which is a very precise genome editing technology. So there are a lot of really kind of incredible tools that are emerging. And some really interesting metaphors that I think connect with making. So this is a paper I'll refer to just from early 2000. This is the Ring Oscillator paper, which is kind of a legendary paper in synthetic biology. I won't explain the details of this, but the basic idea is that the researchers were able to get a cell to produce a phenomenon much like a ring oscillator from electronics. Okay, so this was like one of the first kind of real demonstrations that, hey, we can actually turn a cell into something kind of like a computer. And so synthetic biology, I think in many ways, is an exploration of a metaphor of can we actually engineer a cell the same way we could engineer a computer or engineer a bridge, right? Can we actually take very, very rigorous engineering principles and apply them to living systems? It's a big experiment that is still playing out, but we've gotten pretty far and done a lot of pretty crazy things. So this is an example of a more sophisticated, what's called genetic circuit, which again, this is, I won't get into the guts of it, but there's basically kind of a digital Boolean logic that's running inside this cell. So it's encoded in DNA, put into a cell, and it's running this program. And so a lot of the terminology that's used in synthetic biology is very much like electronics based. So that's why you hear things like genetic circuits. And again, it's an exploration in this metaphor. And so part of what has happened, I was in grad school at MIT when this whole field of synthetic biology was first emerging as a thing. And people, this is a really neat little diagram from a publication connected to a colleague at MIT where, can we actually have a cell that we can sort of really rigorously engineer and work with? And so what are people doing with engineered organisms, right? One giant area is something called basically turning cells into types of factories that can produce all kinds of things, so molecules, fuels, materials. So these are what are called metabolic pathways that nature right now, our bodies right now are producing all kinds of compounds. And so there are all of these metabolites that our body's producing. And so right now there are genetic engineers and synthetic biologists looking at kind of hacking these metabolic pathways to produce all kinds of molecules, right? So cells as factories to make fuels, chemicals. I went to grad school with these guys. These are the founders of a company called Ginkgo Bioworks, which is really the kind of biggest synthetic biology company in the world right now. They're based in Cambridge. And these guys, their whole tagline is the organism is the company, right? So they basically sell organisms that produce stuff. And so clients of theirs span from fragrances to flavor industries, now increasingly into therapeutics, right? So this is a project that I worked on in collaboration with Professor Ron Weiss at MIT. This is basically a cell classifier. It's a little genetic circuit that you put it into a cell, and it does a little computation, and it can tell you if the cell has cancer or not, right? So using microRNAs, actually, some of our colleagues from Tennessee that were doing work with these biomolecules. So there's a lot of things that you can do on the therapeutic side. A big area of research that I care a lot about is something called the microbiome. So looking at the organisms of the body, but this kind of a therapeutic that's being developed now is this idea of a cell-based therapeutic. So microbial therapies, that you can install a cell in the body that could actually be programmed to directly sense and respond to disease. So you don't take any medicine anymore, you actually just install the cell and that thing produces the medicine on demand. So these are the types of kind of things that are moving in the cell and the molecular side. There's a whole other field called cellular agriculture. I'm guessing some of you may have heard about this, so like impossible foods and sort of meats being grown in laboratories. How many of you heard about cellular agriculture before? Okay, most of you, right? And this is, I think, over the past couple of years, just the past few years, we're starting to see more of these consumer products, right? So again, as I mentioned, lab-grown meats, right? Or even materials that could be grown inside a bioreactor. So this is from Suzanne Lee from Modern Meadow, which is a company that works on kind of biologically grown materials. This is actually bacterially produced biofilms, and she was able to make some really cool fashion out of that. Bolt Threads, I don't know if you've heard of Bolt Threads. They're a company that produces spider silk, okay, but inside of a bioreactor. I can't think of something quite more terrifying than like a spider farm where you would harvest spider silk, So fortunately, these guys have figured out a way to actually just engineer simple organisms to produce the proteins from spider silk. So we're seeing all of these applications. This is one of the crazier ones. This is from my colleague at the Media Lab, Kevin Esvelt, who is one of the pioneers of a technology called gene drive technology. How many of you have heard of gene drives before? A few of you. Okay, so gene drive is a very specific application of CRISPR-Cas9. As I mentioned, CRISPR is a technology for very precisely editing, targeting a part of the genome to make an edit. And the idea with the gene drive is that you can actually, using a clever formation of CRISPR-Cas9, through sexual reproduction, pass on a genetically engineered trait through a wild population of organisms. So say you wanted to end malaria in a target local population, and you had an anti-malarial gene in one of these mosquitoes. After multiple rounds of sexual reproduction, you can propagate that gene throughout the entire population. So literally being able to sculpt the ecosystem using these types of tools. So pretty profound things. And so one question I always ask and think a lot about is, who ultimately gets to play? Who gets to make? Who gets to participate, right? How many of you watched the TV show Westworld? A number of you. So Westworld to me is kind of like this dystopian future where it's like the people that have the access to artificial intelligence and biofabrication and synthetic biology, can do crazy things because of this asymmetric distribution of resources. To me, this is a photograph that I took at my community center, EMW, and I think, myself, and I think all of you in this room share a vision where actually, biomaking synthetic biology, it shouldn't just be for the elites. It should be something that is democratized and widely accessible, and that all communities should have a say, should have some kind of agency and participation in this really profound technology. So this is kind of, for me, being here, why it's so important. I think, this whole idea of democratizing fabrication, democratizing making is the same thing that we're doing in the life sciences and biotechnology. And so this question, how do we ultimately do it? How do we ensure we get this broad participation? And ask another way, who gets to make, right? Right now in the life sciences and biotech, who gets to make, right? And right now it is primarily corporations, right? For-profit corporations. Massive biotechnology sector in Boston, where I am. There's giant glass buildings getting erected like on the daily. You walk down the street, and new biotech companies are being built. In academia, of course, places like MIT, which is an extremely privileged place, we get to do things like work with synthetic biology. But part of what's really been exciting over the past 10 years has been the emergence of more grassroots life sciences, right? So do-it-yourself biology or DIY bio was started in part in the US, but I think there's a global phenomenon that happened right around the same time, 2008, 2009. Actually, the first DIY bio meetup took place in this bar in Central Square called the Asgard, which is like this Irish pub, if you can imagine that, being like the start, yeah, Irish pubs, right. So, DIY bio, the first big meetup in the US at least happened there at the Asgard in Cambridge. And, but globally, this has been a really amazing global phenomenon. And so, this is a photograph from, or a schematic from Nature in 2010 and a quote from Rob Carlson, who's a guy that studies the bioeconomy. And he wrote, the era of garage of biology is upon us. Want to participate, right? And so this is, Rob has told me this is his proudest figure. This is him inside his garage lab, surrounded by his PCR machine and his autoclave. And I love how they put in like his little washer dryer over here in the corner next to the broom. So, okay, so now, biology is starting to happen in these unconventional spaces outside of academia and corporations. And so, this is my boss at the Media Lab, Joey Ito. If you don't know Joey, you should look him up. He's a really remarkable guy. He's one of the big reasons why the internet is the way it is. And when Joey first started getting into synthetic biology, this is a picture from about 5 years ago now, we tried to find a lab at MIT where we could get Joey to do some work. And literally, Joey's the director of the Media Lab. He's a pretty powerful guy. And we just got polite no's from everybody. It was like, yeah, we really, really love Joey, but are we going to let a guy that's never held a pipetter before into our advanced tissue culture lab? And the answer universally was no. So we ended up actually doing this first session of biological engineering in his kitchen, right? So we probably violated some of the terms of his mortgage or whatever by doing it, but we did it, right? And so, but fortunately, right, we don't have to do things in Joey's kitchen anymore, in part because of the emergence of these wonderful community labs that now are all around the world. So one of the first labs that was set up in the US was called GenSpace, which is in Brooklyn. BioCurious, Eric Harness over here, I think many of you know Eric, is at BioCurious, which is, I always like to joke, BioCurious and GenSpace are like the Biggie and Tupac of the community lab network, I'm a hip hop guy, so, I like to think about it that way. But, again, really incredible network of these laboratories that's been built up over the past 10 years. Bergen's here from Hive Bio. I highlighted Baltimore Underground Science Space, yeah, Bugs is nearby. So a lot of really wonderful spaces, and again, the projects I think that are coming out of these labs are also really remarkable, in part, I think, because of the incentive structure of community labs versus academia or corporations, right? If you're a for-profit corporation, you got to maximize profits for your shareholders, and you're focused on the next quarter. If you're an academic lab, you're trying to publish papers. And I think the community labs, it's been so inspiring to see the different types of projects that emerge from these spaces. Real vegan cheese, Eric could probably talk a little bit more about this, but I think, you guys were before a lot of the cellular agriculture stuff that happened, right? So, and again, I think a lot of the tools that are very, very low cost accessible, like OpenTron's, Bento Lab, the idea of trying to make a personal computer for biology, these aren't things that academics are thinking about, right? And so, I don't think it's an accident that much like in the personal computer revolution, a lot of these types of ideas of low cost accessible tools are coming out of community labs. And OpenTrons is probably one of the more successful companies that came out of GenSpace that actually now is, like a big venture-backed, company that's doing quite well. But again, I think part of what's so cool, and I'm sure you see in your spaces as well, these spaces, there's an educational focus, there's an innovation focus, but there's also an art and design focus, right? People doing speculative projects about the future of life sciences. So open source gender codes is one that I really love. Glowing Plant, again, which started at BioCurious, was really a provocative project that now, places like MIT are actually running with and doing work with. So anyways, a lot of really wonderful projects all around the world. These are just a few in the US. And again, these spaces being set up in pretty cool places now. So La Jolla building out a space in their public library. There's the BioBus, which I hope one day to visit or have it visit me. Science education on a bus. My colleagues, Jose Gomez, or Jose, who's leads the Little Devices group, setting up maker spaces inside hospitals. Sherry Lassiter mentioned this last time, but our colleague and one of my former students, Ben Juarez, building a floating Fab Labs. So the idea is there's a Fab Lab, but also a bio lab that can engage with indigenous communities on the Amazon, which is really cool. How many of you heard of IGEM before? The International Genetically Engineered Machines Competition? A number of you? Okay, so IGEM is kind of like science or synthetic biology nerd competition, globally and at scale. So 3,000 plus students participate in this every single year. Everybody gets distributed at the beginning of the summer a collection of what are called genetic parts or biobricks. So again, much like engineering, can you build biological systems out of well-characterized parts? And then they basically work on these projects all summer, and then they come to Boston and come to a convention center not like this and share their projects with their colleagues from all around the world. It started out as a college thing. Now there's an overgrad competition, a high school competition. So again, for our colleagues in Tennessee, this is a really cool opportunity to plug in with a global movement here. IGEM also started at MIT, actually. So I was around when the very first IGEM cohort started. And probably my most important role with IGEM over the years is I'm the official IGEM DJ. So I get to kind of rock the big party that happens at the Heinz Convention Center. And it's the one party I DJ every year where I genuinely fear for my life. Because the students are so hyped up. They've been working on these projects all summer. And then the party is kind of the first time they get to really release all of this synthetic biology energy. And it's pretty intense and awesome. So anyways, if we do a NOMCON party sometimes, let me know. I'm happy to DJ it. It's my specialty. Next year, count me in. Yeah, it's my specialty is for DJing for nerds and makers. So let's do it, right? So that's kind of some of the, and I'll spend a few moments now just telling you a little bit about the space that's very near and dear to my heart, which is in Boston, or Cambridge, called EMW. So this space started out, as I mentioned, it actually started out as a Chinese language bookstore, okay? So my parents got the storefront, and they called it East Meets West as a Chinese language bookstore. My father was also a professor at MIT, and he taught electromagnetic wave theory. So his three favorite letters were EMW, for East Meets West, and also electromagnetic wave. So, you know, it's kind of this cultural bridge, and then also, you know, connection to energy propagating throughout the universe. We thought it was pretty cool. So we kept the letters EMW, and that's the name of our community space. And again, we started out doing just poetry, and it was really focused on the Asian American community in the greater Boston area, so it was really a social justice hub. And then over the years, we expanded all of its programming to include all sorts of other forms of artistic expression. You know, again, this is part of the team from a number of years ago. And again, over the years, you know, we've really, I think, made a big impact. And again, primarily, almost all people of color working in our space. Over the past number of years. And again, we've worked with the spoken word communities. This is a lot of local hip hop, and actually all around the country, hip hop artists, electronic music artists. This is Teklan, who's a music producer, the local jazz community in the Boston area. And again, this is MIA's drummer, Kiran Gandhi. Again, beatboxing is a big thing that we've focused on over the years. You know, this is a Nepalese artist who works closely with us over the years. Again, we work very closely as well with social justice organizations, you know, Black Lives Matter, Artists Against Police Violence, and so on are a big part of our space. Street art, salons. This is Juno Diaz at our community library opening, and then of course, you know, biology. So we had our first kind of session around, I want to say 2014 or so was the first kind of sessions that we held where we did some real kind of biohacking in our space. And over the years, you know, we've built out, you know, our laboratory in part with Ginkgo Bioworks, kind of donating equipment. This is full circle for me because I was with the founders of Ginkgo, and we were like Harvard looking for, back when they were looking for equipment. And then I think we got some of the first equipment when they were first, you know, dumpster diving for equipment. So anyways, full circle on that. And so, you know, over the years, we've set up our own space and had a number of different types of workshops and programs, I think like you all do as well. We had a really lovely art program called Culturalizing Science, where we explored the intersection between citizen science and art. And again, we have a really wonderful youth program that I'm very proud of called the Youth Science Initiative, where we work with young people and get them excited about synthetic biology. So we partner with all kinds of folks, including like NASA astronauts, thinking about life beyond Earth and so on, and getting our young people excited about the future of life sciences and biotechnology. Much like you all, we have another big focus on education. So I teach a class called How to Grow Almost Anything. I'm curious how many of you have taken the course How to Make Almost Anything as part of the Fab Foundation? Almost none of you. Okay, that's interesting. So the Fab Foundation, I think as Sherry Lassiter spoke about, they have a really wonderful educational curriculum, and How to Make Almost Anything is a digital fabrication course. And about 4 years ago now, Neil asked myself and George Church, who's this guy over here. If you don't know George, how many of you know who George Church is? Just a few of you. If you don't know George, you should look him up. George is kind of like our modern day Charles Darwin. Like he's one of the key people at the forefront of both reading and writing DNA. So George and I teach this class called How to Grow, which basically explores synthetic biology, starting from biodesign. We teach you how to design DNA, synthesize that DNA, put it into an organism, get it to produce molecules, 3D printing and 3D biofabrication, tissue engineering, all the way to ecosystem engineering. So it's kind of a synthetic biology course across scales. So if you're interested in this class, actually let me know as well. We've taught it globally for a number of years. We taught it at MIT most recently in the spring, but are going to be relaunching a global version. So if you've got a makerspace that's interested in getting involved in the synthetic biology, How to Grow is a pretty cool way to do that. So please come find me if you're interested in learning more about that. But this course, again, it's officially connected to the Fab Lab network. So I always have to update this number. So 1,800 now from Sherry from yesterday, labs around the world. And actually, one of the cool things I'll maybe entice you all with is that I feel like, you know, people that are great at making, like we in the life sciences, like need your help desperately. Like we need to make like lower cost and more accessible versions of basically every tool you can imagine in the lab, because it's really, really expensive still to do work in the life sciences. And Neil has always talked about this vision of like Fab Labs making biolabs, right? Like if you've got a Fab Lab, if you've got a makerspace, you actually have all the tools you need to build your own lab equipment, right? Which is a big, big barrier right now. Increasingly getting smaller, but it is a barrier as a part of quote unquote biomaking. So this course, as I mentioned, we've taught it globally. We have a kind of a virtual classroom and teach it to labs all around the world. Again, this is in my space, EMW. Again, if you'd asked me like, whatever, a number of years ago, from our Chinese language bookstore, would I then be teaching like a distributed biotechnology course, global biotechnology course with George Church, in my Chinese language bookstore, It's really weird sometimes how life plays out, so you never know. But anyway, so this has been a really amazing experience over the past number of years. And again, just a few of the projects that have come out of the class. So things like, biosensitive tattoos. Could you have something that attaches to the skin that responds to different small molecules that come out? Again, there's a lot of fabrication that's involved in biology. So all of the hardware that you can think about, from 3D printers and extruders for biological materials. This is Yishao's project on a living color palette. So taking fluorescent proteins that could be expressed and doing art with that. Yishao is one of our real amazing all-stars that came out of How to Grow Almost Anything. She found our class. She moved to Boston from China by finding this class on the internet and knew nothing about biology. And now a few years later, she's the CEO of a company called Fluidics Bio, which is basically trying to make another desktop biology lab. And it's a $600 desktop lab actually. So if you don't have any equipment and you want to get started, things like Fluidics Bio or the Bento Lab or Amino Labs, which is another type of desktop lab started by one of my former students. Really amazing way to kickstart things. As I mentioned, this year we started globally. We brought it back to MIT, and again, our students there got to experience all sorts of really cool hands-on wet lab experiences. And I think that's one of the best things about both How to Grow and How to Make. Each week we try to teach you, give you a hello world introduction into some aspect of synthetic biology or biotech, be it like microscopy. So this is a technique called expansion microscopy. I don't know if you've heard of this, but this is developed by my colleague at the Media Lab, Ed Boyden, where you basically use the same materials in diapers, essentially, hydrogels, to expand tissues so you can image stuff that normally is so packed together that you don't have quite the optics to actually image. So it's a really amazing technique. Again, we've done microfluidics, 3D biofabrication, cell printing, protein folding, et cetera. And each week we kind of go through one of these topics and teach you something. So these are some of the projects that came out of this year's class. So like a biocomputer and a cream. Hey, could you have engineered organisms that are part of like a skin product that can interact with your skin? And briefly, since this is a maker community, I just want to briefly highlight some of the types of hardware that we're working on in the lab and that we would love to get your help working on with too. So this is the first pipette patent, right? So 1916, how we moved fluids around. This is like 100 years later, and like people are still using the same technology. It's like, it blows my mind that it's still, we haven't had enough innovation, I think, on the fabrication side to make better tools. One thing that I work on though is something called microfluidics or lab on a chip technology, which is, the vision is basically to miniaturize the biolab and put it into a little tiny chip. And so these chips are pretty cool. You can, let's see if it's, oh no, that wasn't supposed to happen. You'll have to believe me that was a really cool video. I'm gonna reopen this. Sorry. While I do this, does anybody have any questions? You can shout them out. I'm just gonna take a second to boot this up. Thoughts? Is this cool? Is this interesting? Yeah. How many of you are excited to build some bio tools in your makerspace? Yes. We need your help. It would be so great I feel like this kind of division of like kind of digital fabrication and software and so on. Hopefully like we can converge on just making, right? Where making is a more kind of universal thing, which I think is happening. Okay, sorry. Let me just and Dorothy, let me know if I'm running out of time. Okay, I'm going to skip that video because that video is problematic. All right, so again, going back to the tooling stuff, so I mentioned the DNA synthesizer earlier. What I did for my PhD was basically building a microfluidic gene synthesizer. So being able to build these gene-like molecules inside one of these microfluidic devices. And so that's cool and all, but you typically need like a semiconductor fab room to make one of these devices because you're casting off of a silicon wafer. So one of the questions I had was like, well, how do we make this lower cost and accessible? And so we published a paper 2 years ago now where we basically built this open source fluidic controller. This is all built off of Arduino. If you go, I'll show a website in a moment, but all of the parts for how to make this stuff is all online and hopefully pretty well documented. And so basically, a number of years ago, I got pretty passionate about doing microfluidics, but instead of just making like super cool cutting edge tools, figuring out how to make them open source and low cost and accessible. And so I started teaching a couple classes at MIT, these open source fluidics classes. I mentioned the Amino Lab, which is another desktop lab. So this is Julie Legault, who founded that company. Julie, before taking this class, had no synthetic biology experience and never picked up a pipetter before. And now she's also the CEO of her own biotech company. This is Will Patrick. Will is a designer, never done any wet lab work, now is the CEO of a company called Culture Robotics, which is one of the hottest companies in Silicon Valley right now. So it just kind of shows you, I guess, in some ways, where we are with biotech, where it doesn't take a lot, actually, for you to get involved in this and really make an impact, if it's in innovation or education, whatever your area is. I'll just share some of the details because this may resonate. So part of what we did a number of years ago, as these commodity 3D printers were emerging, we were asking, well, could you actually make 3D printed fluidics? These are normally, require silicon fabrication. And what we did was, in the context of this class, we 3D printed these fluidics and actually did what's called genetic circuit assembly, so building these big pieces of DNA inside these 3D printed fluidics. And this, again, I think this is what's so exciting to me is like the diversity of the folks involved in biology now and bioengineering. This is a lot of Will's designs. I mentioned Will was a designer. Like, I never would have made structures like this. This is him coming in as a designer, thinking about these 3D fluidics, and made some really beautiful structures. And ultimately, we were able to publish some really cool papers demonstrating the very first 3D printed DNA assembly fluidics. And so, over the years with the course, we've had a lot of fun teaching classes like this. And again, I've worked with other students to not only do DNA assembly, but also things like cell culture, so growing different types of microorganisms inside these 3D printed structures. And again, publishing some pretty neat papers along the way. So one big question, though, and we talked a little bit about this in the session with colleagues from Tennessee, is how do you share all this stuff, right? So one thing that we did, which is something that's active, and I hope you all will join, we have this community called Metafluidics, right? So it's basically this giant repository. It's sort of like Thingiverse, except for biohardware. So you can kind of go to this site, and metafluidics.org, please write it down, and you've got all of these cool little devices. So if you want to make a microfluidic oxygenator, an artificial tooth device, et cetera, you can log onto the site, and all of the design files are there. We could have much better, kind of other support materials like the bill of materials and so on, but we're trying to get better. But I would love for you all, I feel like fluidics is a really great interface actually between making and the life sciences, because all of the hardware that we work with in manipulating these fluids, biology is life. Like, life requires water, it requires fluids, so you need some way to manipulate it. So if you are into making, we would love for you to participate with the Metafluidics community, which is a larger kind of open source global community as well. So there's that. And again, all kinds of crazy hardware you can build. So when I first started my lab at MIT, I challenged a bunch of undergrads. I was like, hey, can you build me a molecular biology lab for under $1,000? And they're like, all right, let's see if we can do it. So they built transilluminators, centrifuges, and so on. And you can get pretty far, actually. And so this is George Church, like, working with some of our low-cost tools. So there's a lot you can do. I'll share with you very briefly a new project that's coming up at our lab, led by Teja, who's an amazing synthetic biologist called Zaphor. And the short version of this is he's built this open source tool that executes a protocol called electroporation. Okay, so electroporation, in short, is like zapping cells so that they'll open up their membranes temporarily so DNA can get inside. All right, so if you want to do any kind of synthetic biology or biological engineering, if you want to recode an organism, you have to get that DNA in there somehow, right? Fine, you printed it in your DNA synthesizer. How do you get it into the organism? One of the major ways is electroporation. Electroporators are typically like a $3,000 or $4,000 tool. There's a variety of reasons why it's that expensive. And what Teja did was kind of amazing. He figured out hey, what are some everyday devices that generate high voltages? And then he basically honed in on the fly swatter, because fly swatters are available globally. He's from India, cares a lot about democratized biotech, and basically figured out how to hack a fly swatter to generate the right voltages and pulse times to make a DIY electroporator. So he basically has figured this out. We're in the process of publishing this paper and getting this all out and sharing all the protocols. So we'll share it with you all as soon as it's out. But he's been kind of going around the world doing these field tests. And again, I'm going to shift to the final section of the talk. this movement's a global movement, right? And people that are involved in the life sciences, you have to get reagents, you have to get tools, and there's a whole supply chain built around that. But if, for my colleagues in Ghana, my colleagues in, Southeast Asia that don't have access to these supply chains, having like open source hardware is like critical to their ability to be involved in the life sciences. So that's sort of why I think the open source aspect is also really important, and why we've also been thinking a lot about how do you kind of hack these global supply chains to make lower cost accessible tools. So the final part of my talk is really about the global movement, okay? So again, when I first joined the Media Lab 2 years ago to start this new initiative, it really was the convergence of these two parts of my life that were kind of separated, right? So synthetic biology and community organizing, and then through setting up our own community lab, we really, I was very grateful to the Media Lab for the opportunity to start this initiative. And so I kind of, I'm from Lexington, which is like, Paul Revere, Revolutionary War, all that, So I kind of was traveling around the world to say, hey, we all as a global movement need to start getting together and working together globally. So I traveled to Switzerland for this event called Biofab and Convergence. There are all of these amazing folks doing DIY bio and biohacking throughout Europe. This was a really amazing event where I got to meet some really marvelous people leading the field there. Folks like Thomas Mboa from Cameroon. Again, people from all around the world, as I think as you all experience in making, look at the life sciences in a slightly different way, right? And so, kind of meeting these colleagues, I think, was for me a very mind-blowing and very profound experience. This is Haquarium, which is a space, a life sciences space in Lausanne, Switzerland. These are all these are all my photographs, by the way. This is from a meeting with the FBI. There's a whole other thing about security we can talk about later. But I kind of spent a bunch of time like engaging with folks that were in all around the world that were doing synthetic biology, but particularly in the community side. So this is Tokyo. These are all different photographs. I love photography, so this is one of my things I really enjoy. Let me see. Oh yeah, so this is Bio Club Tokyo in Shibuya. And basically, I was trying to get everybody together to say, hey, we all need to work together and not just be kind of on our own little silo. And so I organized in 2017 the first ever Global Community Bio Summit, okay? And so basically what this event was, we weren't sure how many people would show up to the first one. We were like maybe 50, 75 people. We ended up with more than 200 people from all around the world that showed up and representing, kind of community labs from basically every continent. And this, I think, event was so powerful because, we've realized, especially in a global context, that you're not alone, right? That you, wherever you are, you're grinding away, you're doing your thing, you're actually part of a bigger global family. And so, we did this thing called Hello World, where basically every community lab got to get on stage and just share for a minute about themselves. And this was like, it was one of the most like amazing experiences of my life, like seeing people from Thailand, from Denver, from Belgium, from wherever they were, from Mexico, Yucatan, just talking about their spaces. And so, this is Joey, and Joey again was a big reason why the internet the way as it is, and has been a part of these technology movements, was asking us, is this a movement? Is this actually a global movement? And the answer, of course, was yes, right? We totally are. And I think one of the things we did in the first Bio Summit that I'm really proud of, and I think something that is still today a very big important part of this movement, is actually engaging in a really intentional way with community organizers. So I don't know if any of you know who this guy is. This is Marshall Gans. So Marshall Gans is a legendary community organizer. He worked with Martin Luther King, with Cesar Chavez in the 60s with the Freedom Riders. He was the architect behind Obama's 2008 and 2012 field campaign. So really rigorously tested this whole pedagogy around movement building, around community organizing. How do you tell stories? How do you actually engage through storytelling? Public narrative is one of the biggest things. So, movements have narratives. They tell stories because they're not just about rearranging economics and politics. They also rearrange meaning. How many of you, I think as technical people and makers, I know for myself and people in the scientific community, we tend to be really terrible storytellers, right? We're really good at making stuff, but it's really hard to communicate like what the stories are, right? And so, working with folks like Marshall, one of the, I think, really amazing things has been injecting into this global community really rigorous community organizing tactics. And so that's what we started in BioSummit 1.0. And then again, much like NOMCON, very experiential type of an event. All kinds of hands-on activities and demos where you can go and engage with bio hardware, do different types of wet lab experiences, et cetera. Of course, awesome party. That's my thing. I love DJing. And and George Church, he's again a big guy in our field, talking about the historic nature of that first event. So fast forward a year later. So this is just about half a year ago now. We had BioSummit 2.0. And basically at this event, we almost doubled in size, right? So we had more than 350 people from all around the world that came and joined the BioSummit 2.0, including Dorothy. So this is like where I met Dorothy for the first time, was meeting her there. I'm so, again, I'm so glad, Dorothy, that you were able to come. And again, I think this connection between our communities around biomaking, community bio, whatever you want to call it, and I think the broader maker movement, I think, is so important. So this event, we had more than 430 people apply, really, really wonderful global diversity. And again, lots of hands-on experiences. We had more than, I think, 30 or 40 different hands-on exercises. We had an art gallery exhibit this time as well. Again, all sorts of wonderful folks like, Drew Endy, who's one of the founders of synthetic biology, but also LaDonna Bravebull. She's one of the leaders of the Standing Rock movement. So we had indigenous leaders there as well. Because again, for me, this is a sidebar, but I really do feel like a lot of the wisdom from indigenous communities is going to play more and more of a role as kind of things move on. I taught a class this semester at MIT called Ancient Modern Technology, kind of looking and critiquing at the way we develop technologies. At the same time, I was teaching how to grow almost anything, which is this hardcore biotech class. So it was a very interesting semester. But anyways, really, really powerful experience again at BioSummit 2.0. And I think the biggest thing that emerged at BioSummit 2.0, to me, I think, is this broader sense of this global community, right? The idea that it's not just about any individual labs anymore, it's about the network, right? To again, I think, riff off of the mission statement, I think, of this particular event. It's about the network. And particularly, we were thinking a lot going into the organizing of this event of, we've got this global community. How do we ultimately organize and structure this, right? And again, here, I've been really influenced by my colleague Tom Malone, who's the father of collective intelligence at MIT. So he basically studies these things called superminds, the idea that when you organize humans together, if they're in hierarchies, democracies, markets, there's some type of a collective intelligence that there could be a supermind. And basically what we're building with this global network, in part through the BioSummit, is some type of a collective intelligence, that there is some sort of emergent behavior that's occurring where the whole is actually bigger than the sum of the parts. And so, if you ask about communities, right, what brings a community together, and I'm sure you all relate with the maker movement as well, in this global grassroots life sciences movement, it's like a bunch of pirate ships, right? Like Hive Bio is doing their thing, Sound Bio is doing their thing, and there is no CEO that's like, hey, GenSpace, go do this or do that, right? Everybody are going to be like, F you, I'm going to do whatever I want, right? So one of the questions is, how do you ultimately bring together this kind of armada of ships? And so we thought a lot about it, and really there are a couple of things, right? So values, having a shared vision, having shared ethics, having shared needs and norms. And so we actually did a bunch of exercises with our community. So we brought in Gunther Weil, who's actually a coach. He's worked with myself and Joey and other people at the Media Lab, who has really rigorously studied values. And we basically had members of our community, about 75 of the whole 430, work on this values inventory. So we could actually try to make explicit what some of the values are for our global community. So things like self-actualization, collaborative individualism, wisdom, right? And I thought one of the key parts about 2.0 was this goal of trying to make explicit some of these implicit things of our movement. Another thing that we did was ethics, right? You can imagine in biotechnology, there are a lot of big ethical questions that we have to grapple with. And so we had a bunch of exercises where our community members could explore an ethical topic and then try to figure out kind of where we as a global community landed. And again, there's more to say about ethics, but this is a big part of what we're structuring for the 3.0 conference. We were trying to build out infrastructure to understand what our global needs were, right? So if you needed something in this part of the world, we have so many colleagues that even though they're geographically dispersed, they actually have very, very similar types of challenges. So how do we kind of connect those communities together and structure resources in a better way? And probably the coolest thing that we did, I thought, was we created a shared statement of purpose. So again, we work with Marshall on this process where we basically developed, starting first with our organizing committee, we generated a version 1 of this statement of shared purpose. Then we workshopped that with groups of around 50 to 75 people, and then ultimately did a larger exercise with hundreds of people all in the same room, and generated the statement of shared purpose, which I will read to you now. So it reads, our shared purpose is to fundamentally transform the life sciences and democratize biotechnology to inspire creativity and improve lives by organizing life science changemakers and bioenthusiasts to build an inclusive global network, cultivate an accessible commons of knowledge and resources, launch community labs and projects, and enable local educators. So what do you guys think of that statement? I don't know. I thought So basically in our closing session, much kind of like right now, we read this statement, and it was an emotional moment. I think everybody was just like so excited, and part of it too, because we made it a co-creation process, people could see like their contribution. It was like, oh, our group contributed the local educator part, or et cetera. So I think that co-creation aspect was something that we really tried to cultivate in BioSummit 2.0. And BioSummit 3.0 is going to be this October 11th to 13th. Applications for the summit, we have everybody apply, although we accept almost everyone. They're open right now, so if you go to, I didn't put the URL on, but biosummit.org, it's super easy. We'd love for you all to come and join. So please, if you're interested in augmenting your makerspace with a biolab, or if you're involved in this movement, please come join us. This is your opportunity to kind of connect with a global family that maybe you didn't know that you were a part of. And so this is 3.0 happening just in a short few months. I'll skip through this. I've talked a little bit about this. We're working on building a platform, community.bio. Anyways, it's just another thing to mention. I will say this. Since the first BioSummit, part of what's really been really amazing is this kind of global movement that's emerged that's connected to other movements. So two of the organizers of what now is a major movement in Africa, the Open Science and Hardware Summit, they met at the BioSummit and were like, hey, we want to do something like this in Africa. And so they organized the first African Open Science Hardware Summit in Ghana in 2018. So this is my first time traveling to Africa before, being in Kumasi. Such a mind-blowing experience. I was a part of the steering committee for the second African Open Science Hardware Summit that just happened in Tanzania. So this is a pan-African event, all folks that care about open science and hardware. And again, going back to the whole global supply chain thing, particularly important, I think, in parts of emerging parts of the world. This is Andrew Quitmeyer. If you don't know Andrew, he's one of the most amazing dudes ever, carries around a whole lab in his backpack. So this was, we did a lot of kind of hacking and making, and this was freaked me out, but actually felt kind of cool once I was doing it. But this is my first time in botanical garden here. This is me with colleagues just basically saying, hey, we're going to build out some community labs in Kumasi in Ghana. Again, folks throughout Europe. And again, part of what's happened now is that there's regional biosummits that are in the process of being organized. So in Europe and Latin America, I spent a lot of time in Southeast Asia recently, in Thailand, where there's a really amazing movement happening there. This is an art gallery, biomaker space in Bangkok. The Fab Cafe, some of you probably know, again, the crossover between, I think, digital fabrication and making. And so this has kind of culminated in something that we just launched called the Global Community Bio Fellowship. So again, taking a lot of our cues from Marshall Gans and leadership development, right? Again, I don't know however many of you think about yourselves this way, but you all are leaders, right? If you're in your community trying to make something happen with a space, like you're a leader. And actually leadership development, I think, is one of the most important things that we as a community can do to try and kind of advance our movement. I'll just briefly pop out of the slideshow to show you, because I'm so kind of thrilled about this. So we just launched this, and our fellows, we have 36 of them. They're from all around the world, every continent, great representation throughout Africa, Southeast Asia, East Asia, Latin America, et cetera. And our goal here is basically to try to find these folks that are setting up labs and really kind of folks that if we can activate and get them a part of a global conversation, could really make a bigger impact. So just really, really thrilled to be a part of all of this. Oh my God, where'd my mouse go? Okay, here. All right. So Dorothy, I'm like out of time, right? I feel like I'm over. Am I okay? All right, then I'll close with one final thing. So we just launched this fellowship program. It's been really, really a remarkable thing. And again, so we basically have these big kind of global Zoom calls where we all log on together and then go through this leadership training where our fellows learn about public narrative, storytelling, how to actually engage with your community, how to structure a field campaign, right? So this is just built off of hardcore organizing techniques developed right here in the U.S. through a lot of the struggles here. So anyway, so I'll conclude now with just an example of a project that came out of our community lab that I think kind of hits for me a lot of my personal interests, but also I think explores the intersection of bio and culture, and I think is an example of the type of project that came out of a quote-unquote biomaker space that you wouldn't really see in an academic or other type of lab. So. And this, I think, again, why is culture important? Innovations don't magically appear or simply proceed on the basis of scientific knowledge. The culture prods them into existence, right? So going back to this idea that the broader public has to be engaged, that this larger societal aspect, I think, is really important to both science and innovation. So there's this field of research called the microbiome. How many of you have heard of microbiome before, microbiome science? All right, almost everybody, awesome. So you've got trillions of microorganisms. Actually, any guesses? If you were to count up the number of cells in your body, like the human cells, and you counted up the number of bacterial cells, any idea what the ratio is, human to bacteria? 1 to 8, that's a good guess. Other guesses? 1 to 10,000. So you're actually, it's something more like, you're about 40%, you're like 60% bacterial cells, actually. Well, it depends though, because most of those cells are in your gut, so a lot of it depends on when you defecate. So that ratio changes a lot at that moment. But anyways, microbiome, one of the hottest areas of science, influences your brain, cognition, sleep, mood, et cetera. But we asked this other question. We're just like, okay, here's the microbiome, very influential in development, but what does your microbiota sound like? What if we could make like a cool project to actually generate sound and music from your microbiome? And so we've made this thing called Biota Beats, okay, which is a microbial record player that translates data about your microbiome into sound and music. Okay, so basically, again, I'm a DJ. This is like DJ scratching vinyl records. This is me scratching a Biota record, right? So all the microbes here. We've got good sterile technique with my vinyl gloves on. And so we literally took a record player, a turntable, and we hacked it and built it into an incubator. And so basically, you can take what are called Biota records, take organisms, inoculate them on the Biota record. They grow, and then we basically image the plate and generate a bunch of data. And then we have some algorithms that convert that into MIDI. And then there's a producer that we work with in LA that turns that MIDI into sound and music. Okay, so this is an example of one of our Biota records. It's literally designed to kind of fit on the record player like an EP or LP record, except it's got media like bacteria food on it. And this is one of our students, Annie, who's an amazing bioartist, sampling some of her toe bacteria and putting it on the Biota record. And basically, you've got these different organisms. They grow over time, and we extract that data, and then we make the music. So you guys want to hear some microbial music? Yeah? All right, cool. All right, so this is, hopefully this doesn't crash. All right, here we go. All right, so this is the sound of some feet bacteria. I'll leave this one. I'll just let you read the captions. This is some belly button bacteria. Your armpit microbes. Oh, and we skipped the oral, but this is what it sounds like altogether. Favorite part, this is the mouth and foot duet. What do you guys think? Pretty cool, yeah? So this project I thought was really, really fun, and part of what I didn't quite realize at the time, and again, real diversity of folks from computer scientists to biologists to hardware people involved in making a project like this. But we thought to ourselves, okay, people have record collections, right? Like, what if you could have like a Biota record collection? Like, what if you could like sample organisms from like really cool artists and make music out of their bacteria, right? So again, I'm a big hip hop guy. This is a photograph I took of Q-Tip. It's one of my favorite rappers and DJs. This is a photograph I took of Questlove. And so I was at this event. I was speaking at this event 2 years ago now with this guy. You know who this is? You guys might recognize him more from this picture. Yeah, so this is DJ Jazzy Jeff, okay? One of the legendary, iconic hip-hop DJs, again, Will Smith's DJ for many years. And so I was speaking at the same event as DJ Jazzy Jeff, and basically I set up a little DIY lab in like in the green room or whatever, and I was like, Jeff, I've got this crazy project. We're making, music out of bacteria. Like, can I sample some of your microorganisms? And, DJ Jazzy Jeff, he's like looking at me, he's like, this is the weirdest question anybody has ever asked me. But yes, I'm down, right? And so this is DJ Jazzy Jeff, okay, like with a little Q-tip. And this is him sampling some of his oral microbiome. And this is, I think, one of my favorite photographs I've ever taken of all time. This is DJ Jazzy Jeff inoculating a Biota Record with some of his oral microbiome. And look at the smile on his face. Look at how stoked he is. He was like so loved this thing so much. And so so we ended up collaborating and making some music out of his beats, out of his bacteria. And probably like one of the greatest moments of my life as a scientific researcher and educator. His management team, called me up like this is a little while ago now, but he was like, hey David, Jazzy Jeff's about to go on tour with Will Smith for the first time in 12 years or whatever, and everybody's kind of freaking out. And she's like, but we've been learning from you that the microbiome is dynamic and changes over time with stress and everything else. So what if we made a Biota Beat of Jeff before he went on tour, after tour, and during tour, and saw how his Biota Beat changed? And I was just like silently crying on the other side of the phone, because this hip hop team, like their management, like they suggested like a scientific experiment, right? Like an actual like multi-data point time course, right? I was like, this thing is working. So I bring this up again because I think music is one of those forms of expression that engages all humans, right, across socioeconomic boundaries, cultural boundaries, political, et cetera. And so we found kind of a way to bring science to people through music, which I thought was really beautiful. And so, so anyways, we got a lot of press coverage, whatever. But one of the coolest things that we did, this was now, again, a little more of a year ago now, we went to IGEM. And we said, could we actually make a song out of the microbiome of the IGEM community? So like 3,000 students from around the world. And so we created this piece called Universe, meaning one song. That's what universe means, our translation of it. And so what we did was we basically wanted to sample the IGEM students, different body parts representing different parts of the world, and basically go through our process to make a beat. And so basically, we had 131 teams that contributed ultimately, and how we set it up was each one of the teams each continent represented a different body part and a different instrument. All right, so we sampled all of the microbiomes from those body parts for those different continents, and then we ultimately created a piece. I'll just play you the very start of it. So this is Universe. So this is first the Latin American teams. And the scalp. So now the African teams with the hands. And one thing too I noticed, the visual is really important, so you can kind of connect the part of the world with the body part in the music. And then the North American teams with the nose. Then the Asian teams where that funky synth bass starts coming in. And so we start to build out with European teams and with Australia. And then the whole composition together. So this was one of the kind of cooler. This is a very intense weekend. We did it basically over 72 hours, where we sampled all the organisms, grew it in the lab, ran the algorithm, et cetera. And these are the pictures of the students that participated. And so it was a really, really wonderful project. And again, I think my closing kind of thing here is just I feel like where we as human civilization, we're facing so many civilization-wide, literally species. Kind of survival questions. And to me, I feel like, we have this incredible global family, and all of this work, I think, is really about trying to crystallize a global community and a global family, yes, about the life sciences, yes, about making, but I think also just about trying to address all of these problems that we need to face together. So, I really hope, and I'm so inspired by being here and meeting you all, but again, I feel like, what we can all do together as a nation, what we can all do together globally, I'm very optimistic and hopeful and part through events like this, so I hope we can all continue to work together to make that happen. So thank you all very much. Really appreciate it.